Welcome back to the deep dive. Today, we're taking a really deep look into something fascinating. The very earliest programming of our development. We're digging into some complex sources, all aimed at finding clear, early life predictors for conditions like ADHD.
Yeah, attention deficit hyperactivity disorder.
Yes.
And that early life focus is just so critical, isn't it?
It seems to be.
I mean, if you look at the theories popping up across psychopathology, the prenatal period, before birth, it's increasingly seen as this really powerful time that shapes future risk.
So we need things we can actually measure back then, in utero.
That's the goal. We need reliable markers from that period.
Okay. So our mission today is zeroing in on one specific kind of novel candidate marker that these sources are highlighting, something influential and crucially something measurable: maternal prenatal inflammation.
Exactly. The big question is, can we actually find a clear biological signal, like an alarm bell during pregnancy that forecasts a child's risk for ADHD symptoms years down the line?
Right.
Because if we can isolate that signal, well, that just opens up entirely new possibilities for maybe prevention or intervention.
It sounds like this knowledge is vital, not just for understanding the cause, the etiology, but also for finding targets we can actually maybe influence.
Yeah, practical targets. The researchers themselves said they're looking for a marker that's easily obtained and low cost for early identification.
That would be a game changer.
It really would. Changes the whole conversation.
Okay, let's unpack this then. Let's start with the big reveal, the main finding from this investigation they did. They followed a group of women, right, from pregnancy onwards.
Yeah, they recruited them during the second trimester and followed them all the way until their children were between 4 and 6 years old. It was a prospective study.
And this long-term approach, what did it show about inflammation?
Well, the finding was pretty novel and specific. They found that maternal inflammation, measured specifically in the third trimester of pregnancy, prospectively predicted a statistically significant increase in ADHD symptoms when those kids were evaluated later.
Wow. Okay, I have to jump in, though. The sources mentioned the sample size was N62 women. Now that sounds, kind of small for such a big conclusion. Should we be, cautious about that?
That's a really fair point. And yeah, the researchers are very upfront about it. A small sample means it's preliminary, absolutely. More studies, bigger ones, are definitely needed for validation.
Okay.
But the reason they likely got such a clear, statistically robust result, even with 62 people, is down to the careful methods. They had the longitudinal follow-up, which is powerful.
Right.
And they used a specific way to measure inflammation, creating that latent variable we can talk about, which boosts reliability. So even though it's small, it's the first human study to prospectively link these specific cytokine levels in pregnancy to later ADHD symptoms in the kids. It's a crucial sort of proof of concept.
Okay, so the fact they found anything significant in a small group might actually suggest the signal is quite strong.
That's one way to look at it, yeah. Potentially.
All right, but if we're talking about a measurable signal, let's get specific. Maternal inflammation still sounds a bit general.
Right.
What exactly were they measuring in the blood?
They were very precise, actually. It wasn't just a general inflammation test. They measured plasma concentrations of 3 key indicators related to a specific immune pathway called nuclear factor kappa B, or NF-κB signaling.
Okay, NF-kappa B.
Yeah. The specific molecules were interleukin-6, or IL-6,
Heard of that one.
tumor necrosis factor alpha, TNF-alpha, and monocyte chemotactin protein 1, MCP1.
Right, okay, that's definitely a mouthful. IL-6, TNF-alpha, MCP1. What do those actually tell us? Why those 3?
Yeah, good question. So think of inflammation as this big immune conversation in the body, right?
Okay.
These 3 chemicals, IL-6, TNF-alpha, MCP1, they're not just random signals. They're like specific distress codes. And they were chosen beforehand, a priori, because research already suggested they tend to be elevated in people who have neurodevelopmental or psychiatric disorders.
Ah, okay, so there's a known connection already.
Exactly. They signal a type of immune activation that seems particularly relevant, potentially, to disrupting brain development.
Got it. Specialized neurorelevant distress signals.
Yeah.
Okay. And how did they combine these 3 different signals into one score for inflammation?
Right. So they used a statistical technique to create what's called a latent variable.
Okay, latent variable.
Sounds fancy, but
Yeah.
it's basically a robust way to bundle those 3 individual signals, IL-6, TNF-alpha, MCP1, into a single, more reliable composite score. a kind of high confidence indicator of the mother's overall systemic inflammation related to this pathway.
So it's not just relying on one potentially noisy measure.
Exactly. It smooths things out, reduces the chance the finding was just a fluke in, say, IL-6 levels alone. It gives you a stronger signal for that underlying inflammation concept.
That makes sense. Okay, so this reliable inflammation score predicts the child's ADHD risk. But what predicts the mother's inflammation? What causes that in the first place? Did they look at upstream risk factors?
They did. And they found some really interesting associations. Three main things stood out.
Okay.
First, and maybe this isn't too surprising, maternal prenatal distress.
Stress.
Yeah, basically. They had a combined score from depression scales and perceived stress scales. And higher distress was significantly strongly associated with higher inflammation levels in the mothers.
So stress physically ramps up the immune system.
That seems to be the connection, yeah.
Yeah.
Stress turns up the volume on these inflammatory signals.
Wow. Okay, that makes a scary amount of sense. What else?
Second, maternal pre-pregnancy body mass index, BMI.
Wait.
Right. Higher pre-pregnancy BMI was also significantly linked to increased inflammation, which fits with other research connecting body fat or adiposity to chronic low-grade inflammation.
Oh, okay. And the third?
The third was interesting, low circulating omega-3 fatty acids, the good fats from fish oil. That was marginally associated with increased maternal inflammation. It didn't quite hit the strong statistical significance level like stress and BMI did, but the trend was there.
Okay, so stress, BMI, and maybe diet through omega-3s.
Right. And, if we connect this to the bigger picture, maybe the most powerful insight here comes from their secondary finding.
Which was?
It seems like maternal inflammation might be acting as a kind of common pathway.
A common pathway. What do you mean by that?
It means it might be the central route, the biological mechanism through which all these different upstream risks, like stress, like BMI, maybe diet, actually exert their influence on the developing child.
Oh, interesting. So like inflammation is the bridge, or maybe the delivery system that takes the risk from, say, maternal stress and actually delivers it to the fetus.
That delivery system analogy is pretty good, actually.
Yeah.
They tested this statistically using mediation analysis.
Okay.
And they found specifically that maternal inflammation mediated the effect of maternal prenatal distress on the child's ADHD symptoms.
So the stress itself isn't maybe the direct hit on the child's brain. It's the inflammation caused by the stress that's doing the work.
That's what the mediation result suggests, exactly. The psychological distress seems to increase the mother's systemic inflammation. And that inflammation is the biological messenger carrying the risk forward to impact the child's neurodevelopment.
That's a huge conceptual shift.
It is. It supports this whole hypothesis that maternal systemic inflammation could be this critical bottleneck, this common pathway for lots of different inputs, psychological stress, metabolic issues like high BMI, maybe nutritional things like low omega-3s.
Right.
And they all might perturb fetal neurodevelopment through this shared inflammatory mechanism.
And you said the mediation wasn't quite significant for BMI and omega-3s?
Not quite statistically significant for the full mediation in this specific small sample, no. But they did observe the indirect effects. The trends were there, suggesting they likely operate through inflammation too. It just didn't clear the statistical bar as strongly as distress did in this particular study. It definitely supports the common pathway idea overall.
Okay. Here's where it gets really interesting for me, thinking about how robust this is. If this inflammation marker is really a key player, they had to check it against the biggest known risk factor for ADHD, right? Genetics.
Oh, absolutely. You have to. ADHD is highly heritable. Family history is like the elephant in the room for any ADHD study.
So did they control for that?
They did, and this is a really smart part of their design. They actually recruited a sample where a large majority, 73% of the kids, had a positive family history of ADHD.
Wow, so they leaned into the genetic risk?
Exactly. They had a high-risk sample in terms of genetics. And the crucial part is, even when they statistically accounted for that strong family history, that genetic loading, the effect of maternal inflammation predicting child ADHD symptoms still remains statistically significant.
Okay, hold on. That's massive.
It's a really important finding.
It means this inflammation signal isn't just like reflecting the genes the child got. It seems to be a separate measurable environmental factor, maybe? Something adding risk on top of any genetic predisposition.
That's precisely the implication. A distinct, potentially intervenable mechanism.
Wow. Okay. And did they check if it was specific to ADHD symptoms or just predicting general problems?
Good question. They did test for specificity. They wanted to rule out that inflammation was just predicting, say, general acting out or maybe the child being moody or difficult.
Right.
And they found that the effect held even when controlling for the child's concurrent negative affect or other externalizing behaviors like aggression. So within this sample, at least, it suggests the inflammatory pathway might have some degree of specificity towards the circuits involved in ADHD symptoms, not just overall behavioral issues.
That adds another layer of credibility. What about who rated the child's symptoms? Was it just the mother's? Because if the mother was stressed and inflamed, maybe her perception of her child was skewed.
Another critical point. That's why a multi-informant approach is so important. They primarily used mother reports, yes, but for a smaller subset of the children, I think it was XT23, they also had teacher reports of ADHD symptoms.
Okay, an independent observer.
Exactly. And even in that smaller group, maternal inflammation levels during pregnancy still predicted the teacher's rating of the child's ADHD symptoms years later.
So it confirms the main finding.
It does. It suggests the effect isn't just an artifact of biased maternal reporting. That consistency across different raters really adds significant weight.
Okay, this is building a strong case.
Now, what's fascinating here is getting to the biology. How could the mother's inflammation during pregnancy actually predict neurodevelopmental risk in her child years later? I mean, it's not magic, right? There has to be a physical link.
Right. How does that inflammation delivery truck, as you called it, how does it actually get its message across the placental barrier?
Well, there are several ways this is thought to happen. The sources outline about 4 major potential mechanisms.
Okay, let's hear them.
First, the maternal cytokines themselves, those IL-6, TNF-alpha signals we talked about. Some of them can physically cross the placenta.
Really? They get into the fetal environment?
Yeah, potentially leading to higher cytokine levels, not just floating around, but maybe even directly in the fetal brain tissue itself.
Yeah.
Basically bathing the developing neurons in these inflammatory compounds.
Wow. So the fetus is literally directly experiencing the mother's inflammatory state.
That's one possibility, yes.
Sure.
Second, these maternal inflammatory signals can trigger epigenetic changes in the placenta.
Changing how genes are expressed in the placenta.
Exactly. The placenta is this incredibly powerful regulator of fetal development, kind of programming the fetus. Inflammation could essentially reprogram the placenta to put the fetus into a sort of high alert stress state.
Okay, that makes sense too. What else?
Third, the signals can activate the immune cells that reside right there at the boundary, the maternal-fetal interface. Kind of stirs up the local immune environment.
Which then probably leads to cascade effects.
Yes,
Okay.
that's the fourth point. All of this, the direct cytokine crossing, the placental changes, the local activation, it can prompt a significant inflammatory response within the fetus itself. The fetus mounts its own response.
Oh.
And that fetal inflammatory response is what's thought to directly shape or maybe perturb brain development. It could affect how neurons connect, how circuits are pruned, things that later manifest as behavioral differences like ADHD.
Okay, that biological cascade makes the link seem very plausible. So this moves us towards the practical side. If this finding holds up, if it's confirmed in bigger studies, what's the so what? How could this actually be used?
Well, the hope is that this kind of finding really sets the stage for developing and testing interventions. For clinical trials aimed at reducing risk in people who might be susceptible.
How would that work?
If you could identify women with unusually high inflammation early in pregnancy, maybe through a simple blood test, perhaps you could intervene. For example, the sources mention anti-inflammatory interventions given prenatally, like potentially omega-3 supplementation.
Ah, the omega-3s again. You said low levels were linked, maybe weakly, to higher inflammation. So the idea is boosting them could lower inflammation and maybe lower risk.
That's the hypothesis. And there's some backing for it, at least from animal models. Studies in mice have shown that giving anti-inflammatory treatments during pregnancy can actually lessen the impact of prenatal stress-related inflammation on the offspring's hyperactive-like behaviors.
Interesting. Has that been tested in humans for ADHD prevention?
Not rigorously, not yet specifically targeting ADHD risk based on measured inflammation, as far as these sources indicate. But the biological pathway is there.
Yeah.
If inflammation is the delivery truck for risk, maybe something like omega-3s could act like a roadblock, or at least lessen the load. It's an avenue for future research for sure.
Okay. Now, before we wrap this deep dive, we absolutely need to circle back to the cautions. We talked about the small sample size, N62, preliminary needs replication. What other limitations did the researchers point out?
Yeah, besides the sample size, they highlighted a couple of other important ones. First, they only looked at those three specific cytokines, IL-6, TNF-alpha, MCP-1, all related to that one NF-κB pathway. But inflammation is way more complex than that. There's a whole orchestra of different inflammatory molecules. Looking at a broader panel might give an even clearer picture.
Sure, you might be missing other important players.
Exactly. And second, they only measured the inflammation at one single point in time during the third trimester.
Ah, okay.
It's very possible, even likely, that the timing of inflammation across the whole pregnancy matters just as much, if not more, than the level at one specific snapshot. Maybe second trimester inflammation has different effects, or chronic inflammation versus a short spike. That needs way more investigation.
Those are important caveats. Still, despite those limits, you mentioned the strengths earlier, the longitudinal design, following kids over years.
Right.
Using multiple informants, like teachers, not just moms. And that careful statistical approach with the latent variable to get a reliable inflammation score.
Yeah, the methodology itself seems pretty sound, which is why these initial results are generating excitement, even with the small n. It's compelling.
So let's bring it all together. What does this all mean? What's the big takeaway here?
I think the main takeaway is that maternal prenatal inflammation, specifically linked to these cytokines, is emerging as a really novel and potentially very promising biological marker for risk of child ADHD symptoms.
Okay.
It looks like it could be a core biological mechanism, the common pathway idea again, through which lots of different seemingly unrelated prenatal risks, like psychological distress or high pre-pregnancy BMI, actually transmit their influence to the child's developing brain.
So it turns something kind of vague, like stress during pregnancy, into something potentially quantifiable, biological, and maybe, just maybe, actionable.
That's the hope. That's the potential power of identifying these kinds of mechanisms.
All right. And maybe here's a final provocative thought for you, our listener, to chew on. This study linked maternal BMI and prenatal distress to this inflammation pathway, which then predicted ADHD risk. We know inflammation might be this common pathway. So what other factors, things maybe not even psychological, like what about a serious infection during pregnancy, or maybe undiagnosed chronic health conditions in the mother, or even exposure to certain environmental toxicants? Could they also be funneling risk through this same inflammatory common pathway to shape brain development?
That's a huge question.
And if we could map all those different inputs that trigger inflammation, how much earlier, how much more comprehensively could we potentially intervene to support healthy neurodevelopment? That seems to be where this line of research could be heading.
Absolutely. Identifying all the inputs into that common pathway and understanding the timing, that's the challenge for the future.
Definitely food for thought. Thank you for diving deep with us today on this really important topic.
My pleasure. It's fascinating stuff.
We'll see you next time on The Deep Dive.